Hello students, this is Dr. Anita Raj, your chemistry mentor, welcoming you for another session in Biomolecules. Today we are going to see about proteins, specifically amino acids, peptides, peptide bonds, Zwitter ions and isoelectric point. Okay, we shall move on into the topic right now. So now we shall see about proteins. See, the word protein is derived from the Greek word proteus, which means primary or of primary importance. Okay. See, actually, these proteins constitute about 19% of the average human body and serve several functions in the body. Actually, they are the main constituents of a living cell and are essential for the growth and maintenance of life. Okay. See, these proteins are highly complex nitrogenous organic compounds of very high molecular mass. So these proteins are highly complex in nature and they are nitrogenous organic compounds, and of course they have high molecular mass. Okay, and they are the polymers of alpha amino acids and are connected by peptide linkage. What do you mean by peptide linkage or a peptide bond? Actually, it is a new bond. See, it's a new bond. C double bond O NH. This is formed between two or more amino acids. Okay, these are formed between two or more amino acids due to condensation. Okay, so these peptide linkage are formed by the condensation of two or more amino acids, right? So this is called as peptide linkage or peptide bonds, right? So what are amino acids then? See, these amino acids are the building blocks of protein. Okay, they are the basic building blocks of proteins. Actually, they contain the amino group, NH2 group, and the carboxylic group, that is COOH. So these two functional groups are present in this amino acids. Okay, the simplest amino acid is glycine, and its formula is NH2, CH2, COOH. See here we have both the functional groups, NH2 group as well as the COOH group also is also there. So except glycine, all the rest are optically active. What do you mean by optically active? When do we call a compound as optically active? We call a compound as an optically active compound when it rotates the plane of polarized light. Okay, if the compound has the capacity to rotate the plane of polarized light, then we call that as optically active. So except glycine, all the other amino acids are optically active. Okay, so alpha amino acids contain alpha carbon atoms. What do you mean by alpha carbon atom? So this is the first carbon atom to which the functional groups are attached. Okay, so alpha amino acids are nothing but those amino acids will be having alpha carbon atom. So this alpha carbon atom, the functional groups will be attached and are asymmetric in nature. Okay, they have the chiral center, they are asymmetric in nature and they exist in two stereoisomeric forms, D and L. What are stereoisomers? Stereoisomers means they'll be having the same molecular formula but will be having different spatial arrangements. Okay, so D and L. Okay, these two forms, D and L forms, are the mirror images of each other and are also called as enantiomers, and they are also called as enantiomers. Let me write the example for alanine. Alanine is an example of amino acid which has a chiral center and it is optically active. Let me write the D and the L form of alanine. C NH2 COOH CH3 and H. See, this is the D form. Okay, D-alanine. This one is D-alanine. In L-alanine, this NH2 group, okay, this NH2 group which is present on the right hand side will be seen on the left hand side. Can you understand? That's the difference. So here we could see the NH2 group. Okay, NH2, here H, this side COOH and CH3. So this is L form of alanine. So depending upon the number of amino and carboxylic groups, amino acids are of three types, right? So we are going to see the classification of amino acids now. Now let us see the classification of amino acids. We are having three types: neutral, acidic and basic. See, in neutral category, it contains equal number of amino and carboxyl groups. Okay, in neutral amino acids, we have equal number of amino and carboxyl groups. See glycine. See here, this is amino group, this is carboxyl group. Equal number, we are having 1, 1. Alanine, 1, 1. Again, leucine, 1, 1. So equal number of amino groups as well as carboxyl groups are present in neutral. Whereas in acidic, it contains more number of carboxyl groups compared to amino groups. See, in aspartic acid is an example for acidic amino acid. Two COOH groups are there and but only one amino group. In glutamic acid, two COOH but only one amino group. Is it not? Next, see the basic. It contains more number of amino groups compared to carboxyl group. See here in lysine, 2 NH2 and 1 COOH. In argine, 2 NH2 groups. So 2 NH2 groups and 1 COOH. So with more number of amino groups than carboxyl. So these are the different categories of amino acids. Okay. So next classification is based on the availability. Okay. So the first one is essential amino acid. The next one is non-essential amino acid. Right. So what are essential amino acids? Amino acids which cannot be synthesized in the body and must be supplied from outside. That means through diet are called as essential amino acids. Okay. So in our body we will not be having this type of amino acids. Essential amino acids are not available in our body directly. So that has to be supplied through other supplements, either through diet or through pills, etc. So these type of amino acids are called as essential amino acids. And example of essential amino acids are valine, leucine, isoleucine, lysine, arginine, histidine, phenylalanine, methionine, tryptophan, threonine. Okay. So these are the different types of essential amino acids. Next category is non-essential amino acids. Amino acids which can be synthesized in the body are called non-essential amino acids. See, these amino acids are already present in our body that can be synthesized by our body, prepared by our body. So those amino acids are called as non-essential amino acids. Okay. Example, glycine, glutamic acid, aspartic acid are examples of non-essential amino acids. Okay. So next let me explain about the peptides and the peptide bonds. So already I have explained about peptide linkage or peptide bond. But let us see once again what are peptides. See, the compounds formed by the condensation of two or more or same or different alpha amino acids are called as peptides. When two or more amino acids or same or different types of amino acids condense together, we get the peptides. Okay. So that new bond C double bond O NH formed between two amino acids due to condensation is called as peptide bond or peptide linkage. So this is the bond, okay. This type of bond is formed between two amino acids. That is called as peptide bond or peptide linkage. Understood, students? Next, we shall see the important thing, that is the properties of amino acids. See, these amino acids are usually colorless. They are crystalline solids with high melting point. Okay, these amino acids are colorless crystalline solids with high melting point. Okay, they are water soluble. They are soluble in water and behaves like salts rather than amines or acids. Okay, they behave like salts since they are soluble in water. They behave like salts rather than acting as amines or acids. Okay. This is due to the presence of both acidic and basic group. Why this is soluble in water? Why this is behaving so? Since because this amino acids, they have both acidic as well as basic group in the same molecule, which is called as Zwitter ion form. Now let us see what is a Zwitter ion. So actually in aqueous solution, the acidic carboxyl group, that is COOH, which is present in the amino acid, loses a proton to the basic amino group NH2 group to form a Zwitter ion. See, this is an amino acid. NH2 group is there. This is an alkyl group CHCOOH. What is happening here? The proton which is present in the carboxyl group will be moving to the NH2 group, amino group. Okay. So that this gets a positive charge. Okay, NH3, it becomes NH3 plus and this becomes minus. So this is a neutral compound now. We have both plus and minus. It is dipolar. Okay. So this is called as Zwitter ion. It is called as dipolar since it has both positive and negative charge. Since because a proton is moving from the carboxyl group to the NH2 group, this becomes positive and this becomes negative. Okay, since it is losing a proton, it is becoming a negative. So and it's a neutral compound now and it is called as dipolar. Okay, and the other name for this one is Zwitter ion or internal salt. What is internal salt? Zwitter ion is otherwise called as internal salt. This is the reason why the amino acids are behaving like salts. Okay, they are water soluble. Understood, students? Now let us see the characteristics of Zwitter ion. Actually, it is a dipolar ion. Obviously, it is correct because it has both the charges, plus charge and minus charge. It is neutral in nature. Yes, obviously, it is neutral since because it is having plus and minus charge. It shows amphoteric behavior. Yes, it is. What do you mean by amphoteric? Amphoteric means it acts as acid as well as a base. So it shows an amphoteric behavior. Due to dipole-dipole interaction, they are having high melting point. due to dipole interaction. They are very strong enough. The bond is very strong enough, and it's not easy to melt this amino acid. So it requires high temperature. That's why it's having high melting point. Why these amino acids or the Zwitter ions are having high melting point? Due to the strong dipole interaction. Can you understand? Now let us see what you mean by isoelectric point. This is very important. See, actually in acidic medium, I'm speaking about the Zwitter ion. Okay, so there's a strong relationship between the Zwitter ion and the isoelectric point. See, in acidic medium, COO minus group, which is present in the Zwitter ion, acts as a base and it accepts a proton. Okay, in acidic medium. Okay, what happens in acidic medium? The COO minus, which is present in the Zwitter ion, will be accepting a proton. It acts as a base and it accepts a proton. Okay, so what happens? This is accepting a proton. Okay, since it is accepting a proton, this becomes OH. This becomes OH and we have a positive charge here. Okay, since because it is accepting a positive charge, this ion, Zwitter ion, which is a dipolar ion, now becomes a positive ion. Okay, because it is getting neutral, this is neutralized, so it's becoming a positive ion. Since it is having a positive ion, under the influence of electric field, this positive ion will be moving towards the cathode. Okay, understood. As a result, the alpha amino acid exists as cation in the acidic medium. It is existing as a cation in the acidic medium and migrate towards cathode under the influence of an electric field. Can you understand? Now in basic medium, just the reverse is taking place. What happens is the group acts as acid and gives a proton. Now this gives a proton. Okay, this gives a proton. In basic medium, this Zwitter ion will be giving a proton. So one H plus will be given to this base. Okay, so what happens? This OH minus will be reacting with H plus and we'll get water. So there is removal of water. Understood? So now what happens? Since this NH3 plus is giving one proton, this NH3 will become NH2. Now NH2 CHR, CHR, C double bond O, C double bond O minus. So now this is becoming a negative ion. Moving under the influence of electric field, this will be moving towards the anode. Can you understand? Under the influence of electric field, this will be moving towards the anode since it is acting as a negative ion. Can you understand? At some intermediate pH, when the pH is changing, the concentration of cation and anion becomes equal. Okay. So in acidic medium, it is acting as cation. In basic medium, it is acting as anion, the Zwitter ion. But at a particular intermediate pH, both cation and anions becomes equal, and no net migration of alpha amino acid takes place in an electric field. There will be no movement of alpha amino acid taking place in an electric field. Can you understand? This pH is called as isoelectric point. So this pH is called as isoelectric point, and the amino acid will exist as Zwitter ion with maximum stability. So at which pH or at which point the Zwitter ion will be having maximum stability? In isoelectric point. At the isoelectric point, the Zwitter ion will be having the maximum stability. That means it will not be moving to any of cathodes or anode. Understood? Well, fine students, I hope you might have understood what I have taught today. Let me meet you with another important topic in my next session. Until then, it's Dr. Anita Raj, your chemistry mentor, signing off from it. Thanks for watching.